All right, we're in the final stretch, everybody. Welcome back for this last half-day session. Thank you for sticking through. This morning, we'll be focusing on system suitability for LC-MS. I'd like to take a poll. How many people run system suitability samples or something like that to make sure your LC-MS integrated system is working as you expect? Awesome. If you were to come back to this course next year, I would hope to see all of your hands raised, because this is one of the simplest and smartest things that you need to do to be able to trust the data that you generate on your real samples. Okay, so system suitability is a kind of a very boring topic, but fortunately I like really boring things, and so I've spent a lot of time developing system suitability and evaluating it and helping to try to move the field to also do the same for proteomics data. Okay. First, what is system suitability? And here are a couple definitions that I pulled from the interwebs. One is from the FDA. One is for this International Conference on Harmonization of Technical Requirements for Registration of Pharmaceutical for Human Stuff. And these are very wordy definitions. Simply put, it's the analysis of a known sample to assess your system performance. Many people don't think this is necessary, or the thought doesn't come to them, because we calibrate our mass spectrometers to make sure we have accurate mass, isolation, detection intensity, but we do that by infusion of a calibrant. We might be able to prime and purge, measure the flow rate of our LC, and that's great, but that doesn't really take into account the column that we use, which is integral to LC-MS. So the idea here is we want to take our system that we're using in its entirety to run our samples, monitor a known sample, track its performance over time, and hopefully predict when the system might be beginning to go out of performance criteria that is acceptable. Also to monitor its performance and define an acceptable range of retention time shift or peak area variability. And so this is meant for illustrative purposes, but it is real data. When we first started our CPTAC studies, not every lab was running a system suitability. So when we did our second study, we insisted that everybody should. And here we've got one lab that was running, and all of the transitions overlay, and it looks beautiful, and the peak width seems it's a nice Gaussian shape. And then we had a lab that had never done nano flow before, and they were having problems. This was before the Nano Viper fittings and a lot of the easy to put together nanoflow fittings were available. And so people were cutting fused silica and doing their best to get it together, and their peak shape looked like this. And they didn't realize that that was not good, because they were new to this whole process. And so you can see here, this peak width is less than 30 seconds, and this one is like 2 minutes. So it's always a good idea to help you have confidence in your system performance and the data you're producing with it to do system evaluations. Some people use QC as a term for this. I use system suitability because it's evaluating the whole system and not just aspects of a workflow, which is what I would use QC for. Okay, leading in, is system suitability the same as QC? I don't believe it is, but there may be some debate out there. QC is quality control of your method. So if you spike an intact protein into your sample, you are doing a quality control on your digestion. We're not looking at our sample handling workflow. We're only looking at performance of our LC and MS system integrated together. QC takes into account the sample prep. You can add in low, medium, and high QC standards to your entire experiment to help make sure that the process your sample's going through is as expected, and that the detection of your samples is as expected. And so, again, relying on expected responses. QC is method specific. You're not going to run a small molecule QC if you're doing peptide analysis. and you're not going to spike in a protein that has nothing to do with your sample. Yeah, for QC, that would be something that wouldn't work very well. System suitability is designed for long-term system evaluation. It is not a new concept. It's been used through the decades and many, many decades in all sorts of analytical techniques, process control. Questions so far? Okay. When should you run a system suitability standard? I say all the time. I don't trust anything until I make sure my LCMS system is running as expected. My retention times are where they should be. My peak areas are within a certain range. So I will run it before running unknown samples, obviously. Periodically, many times a day, one time a day, it's kind of up to you how long that system suitability run is and how much time it takes away from your sample analysis, keeping in mind that when you have a run that fails system suitability, everything before that is suspect. So if you want to run a system suitability once a week, then you have the option to potentially throw out a week's worth of data that you're not sure if it was performing correctly. Before and after instrument repair, usually we run a system suitability to say, oh, something's wrong, call an engineer in, they fix it, you run it after, hopefully everything's back to normal. Sometimes it's not. Before and after changes to hardware or plumbing. If you change a column, if you change tubing, capillary tubing that many people use doesn't come inherently labeled once it's cut. It just has the same outer diameter. And so unless you have a microscope and go and look at the inner diameter, or you're very diligent and label the inner diameter of your tubing, it's easy to swap out something that's 200 micron ID with something that's 50 or 20. And that will have major impacts for nanoflow. Also, if you have kinks or leaks in your system, system suitability will pick it up. Before and after changes to software, right? So I didn't realize how important this was until I worked for Thermo and realized that a lot of the things that control mass spec performance are heavily reliant upon the software. And bugs can cause issues with detection, issues with isolation. Incorrect voltages can be set, so ions aren't going through and being detected. So this is hugely important. If a vendor says, hey, we want to upgrade your software, it's got all these great new features, just be like, hold on, let me run a couple system suitability to make sure before and after it doesn't get worse. Anytime you think your system is not performing optimally is always a good time to measure and see if it is. And then when you think your system is performing optimally, it's always a good idea to get data so that you can look at it longitudinally. And we have people that have done evaluation of system suitability to help predict when a system might begin to go out of control before it actually does. So you can intercept, do whatever little changes need to be done to help get it back into your performance criteria before it really goes off and major repairs or changes need to be done. Okay. So what do we look at? What are the metrics of system suitability? We look at, so we break it down into two major parts, MS and LC. For MS, we look at the response, the sensitivity, the reproducibility of that, the background, the noise. This can change based on things in the atmosphere. I worked in a lab once that had construction of a building going on, like just outside the windows, and we started to see elevated noise because of all the additional dust and things that were in the atmosphere. Remember, mass spectrometers don't just accept ions into their orifice. It's got a vacuum that sucks in anything. So I've seen pictures of a mass spec that sucked in a spider or a mosquito. Like, it truly happens. So dust is something that can definitely enter the mass spec and cause background noise. Looking at your fragment ion transition ratio, whether it's full scan or SRM, as long as you're above your limit of quantitation, this should be consistent all the time unless you have interference. And then the precision of all these measurements. For the LC, you want to see reproducible retention time. Nanoflow is always tricky, and there are a lot of things that affect the reproducibility of the retention time, but the point is you want it to be as reproducible as possible. When you see shifts of minutes, that's something that maybe isn't something you should want to accept. Peak shape, full width, half max. Chromatographic peaks should be Gaussian shaped and like equal. equally shaped on both sides. Some analytes will have tailing, right? Or if you have isoforms of a certain form of a peptide, you might see a humpy, not well-defined peak, and that happens sometimes. But the majority of the time, you should have nice Gaussian-shaped peaks. In the previous example, where is it? Yeah, this was just, oh boy. This was just bad fitting, which caused post-column band broadening. And so that's not normal. That's something that can be fixed. All right, chromatographic resolution. This means the separation of peaks as they elute. If you see that your peaks are all nicely separated and then over time they begin to coalesce and broaden and overlap, that also is an issue and something to pay attention to. And then carryover. This is obviously something you don't want to happen if you're trying to quantify low abundant things in your sample. So overall, the goal is to confirm that you have adequate signal to noise, and all the reproducibility is what you expect before you analyze your real samples. So chromatographic resolution, this is, when we talk about LC-MS, most people think resolution is like the Orbitrap or the TOF, and like how well you can get highly accurate mass on your mass spec data. But chromatographic resolution is something that is equally as important. If you can imagine, you run a sample in your system suitability and you've got these two peaks. You can calculate resolution very easily with the data that Skyline provides by retention time and full width half max, and get a value for chromatographic resolution. We can do this on two adjacent peaks or peaks at the beginning and end of a gradient if you run the same gradient for your system suitability, and you get a numerical unitless value. If that changes significantly, then obviously something's wrong either with the column or the LC pump or the connections. And if you define an acceptance limit, this is something that you can always refer back to. Okay, this was a paper that came out almost a decade ago, wow, where part of the CPTAC study was to evaluate the technology for defining candidate biomarkers in cancer. And all of the technology, not just for quantifying. So there were a number of labs that participated in this study, and they used for discovery proteomics. And the NIST was involved, the National Institute of Statistical Standards, and they broke down all of the possibilities for potential variability or things that you could measure into categories. And this used to be, I'm not sure if it's still available, I'll have to check. This used to be available online where you could go and take your data in, and you would be able to get measurements of all of these different criteria that could affect the outcome of your data, whether it was, if it was data dependent. Any questions so far? Okay. So hopefully those of you that have not been using system suitability are now like, oh, this does sound like a good idea. Where do I start? What do I do? It's never too late to start. Always start when you can start. But the question might be, what can you use as a sample? The idea is you want it to be something that's similar to what you're analyzing routinely on your instrument. I realize some labs, like ours here, may ping pong between doing peptide analysis and small molecule analysis. And in that case, you might have two different system suitabilities that you monitor when you're doing those specific analyses. So you'd like them to be similar to your actual samples so that you can sort of extrapolate if the performance is going down, how it might affect your actual samples. Something with minimal carryover, oh, back up here, a stable sample. You want to make sure that if you see changes in the data outcome over a period of time, it's not because your system's degrading or your sample's degrading. You want to make sure that that sample is solid. You want to make sure it has minimal carryover. For example, if I'm doing a lot of human studies, I wouldn't want to use digested plasma as my system suitability because it's super complex, and also it could carry over and affect my real samples. Something that if you have to prepare it is very easy to prepare. Ideally a mixture of several candidates. Some people may use beta-gal as like a retention time evaluator. It's one peak, right? Right, it's just one, I think it's one analyte. So like, what do you do with that? you can't do chromatographic resolution. You can do mass accuracy and peak shape, but like, wouldn't you want to have a couple other measures in there? Ideally, commercially available in QSeed. So Promega, Sigma, Pierce have all spent a lot of time looking at diagnosis, have all spent a lot of time looking at potential standards that you can use. Some of them are expensive. To be honest, it's quite worth the investment because a couple $100 on a standard that lasts you 6 months is way better than running 6 months worth of data and not having anything to show for it at the end because your system wasn't performing properly. So some examples are cell lysate digests, digested in bulk and aliquoted and frozen until use. And so yeast or HeLa, you can do this yourself if you do them in large enough batches and make sure that when you change from one batch to another, their performance, you run them both interweaved so that their performance can be accepted as parallel. Synthetic peptide standards, so these are the Biognosis, the Pierce retention time standards, the Promega 6x5. A simple protein digest, some people use BSA or, what's the other one? There's one that Thermo gives you when they install a new system. I forget what, Cytochrome C, that's right. NIST standards, so if you really don't want to mess around, there are standards that NIST has qualified using a variety of techniques to make sure that the concentration is what it's supposed to be and so on. Isotopically labeled compounds, these are usually preferred because they will not technically interfere with, they're not supposed to interfere with a proteome sample because they are isotopically labeled. And then peptides spiked into a complex background. So my go-to is a cell lysate digest like HeLa lysate with retention time standards spiked in. I usually have the HeLa lysate digest at about a microgram or 500 nanograms on column, and then I spike the peptides in at a level where I know I should get reproducible peak intensities. Not so high, but something that's measurable, and that if the sensitivity of the system declines, I will see that effect as an increase in the variability of my peak areas. Okay. How can you assess normal system performance? So if you're starting from scratch and you've not done this before, first tune and calibrate your LC and MS components. Follow manufacturer's recommendations, obviously. Particularly with the LC, this is a part that is often overlooked because it's got solvents in the columns and it's full of solvents. It should be fine. Most of the time, particularly with NanoFlow, it's not. You have to baby them. You have to make sure your solvents are fresh and degassed. Don't underestimate the effect that that will have. I was involved in a study last year where we weren't getting reproducible system performance when evaluating a new piece of technology. And of course, we blamed that technology and not the LC. And when we went back and said, oh no, we have to prime, use degas solvents, purge, flush the system, measure the flow rate, oh my gosh, now everything is working, imagine that. So don't underestimate making sure your LC is working well. Don't top off your buffers either. I used to do that and like laugh in the face of engineers, and now I have been humbled. Monitor the variability of these metrics over several injections. So if you do 10 injections all at once, all at the same time, it gives you variability over a very short period of time, and so that variability should be small and good. If you do it once a day over a course of a week, you might expect the variability to be a little bit higher, but it's always good to assess it, and it's not difficult to get the data. Ask the vendor for performance specifications for both LC and MS parameters. A lot of the vendors are now beginning to come up with methods, with standards that you just get an app note or something very similar and just plug it in and do it. They know what performance to expect. You can make sure that that's what you're seeing, and if not, you can either go back and say, well, did I do something wrong with the method, or do I not have the column connected right, or evaluate what might be causing the changes. Note, obviously, when the system performance deviates, and try to troubleshoot what might be causing that. Sometimes it's simple, like an old column, or your solvents are not degassed or your system's not purged, or your mass spectrometer has not been calibrated and the mass accuracy is off. And then this is a luxury, but compared to other systems running the same system suitability protocol. So like there are 40 some of you here. I'm sure there are a couple people that have the same type of LCMS system. Connect, start running the same type of sample, and then compare data. Because that was a big thing we saw with the CPTAC study was labs that were isolated and just running their own stuff in their lab didn't realize when the system wasn't working well because it was good enough. Well, when they compared to another lab that was getting twofold better intensities, they're like, hey, what's going on? This isn't right. And so you can do that here, like network and talk to each other and build connections because that'll help strengthen your data and your science. How can you evaluate system suitability for free? There are ways in software that you can use that if you're working for a pharmaceutical company or a place with an enormous budget, there may be things that are already in place and available. When I started this, we didn't have that. There used to be this executable software called raw meat that was specific to thermo that would take, for example, your data dependent data and pull out over the course of the gradient how many MS scans were performed, how many MS2s were performed, what was the average injection time, and details like that. This has been upgraded to software called Tartar. Get it? And so it takes the data and pulls out any kind of numerical information it can get out of it so that you can look at that and pull what you need and track it over time. The NIST metrics website, Skyline, SPROCOP, Panorama, and AutoQC are all something that have evolved over the years, and now it's become a pretty well automated process. Retention time viewer, this is something that was developed for the CPTAC labs where we could take everybody's data from all the different labs and look at what the retention time was for each of the peptides in the sample and see how far they off for, in this system versus this other system that was several states away. Database searches for peptide ID, this is okay. It's not ideal. A lot of people, when they do data dependent runs, will get a number of peptide IDs. Like, that's okay, but it's not solely by itself something that I would recommend using. You can use Excel. You can put in retention times. Totally manual and a pain in the butt, but it still works. Using any longitudinal data tracking system, or check with your instrument vendor because some of them might have something in place that it's just not advertised or talked about because, again, this is a kind of boring topic. So qualitatively, we can review the system suitability data. Here is an obvious, like, whoa, something's totally wrong there, and it's not always obvious, especially if things are slowly changing over time in a systematic way that will eventually drift out of instrument performance. So you want to use predefined quantitative acceptance limits that you can set up for your system. So Skyline has a lot of nice features that allow you to kind of look at the data from a high level and say, like, oh, well, for this one run, we've got great peak shape, but I can look at the reproducibility of the retention times across 10 injections. I can see that injection 1 eluted a little later, and then they kind of drifted earlier and kind of stabilized, maybe going back a little later. Some of it can be explained, like a new column might have later retention times because it hasn't been conditioned yet, and your peptides might linger a little longer interacting with the stationary phase, and their retention times might be a little later. And then as it's conditioned, it will stabilize. If the column's aging, you might see it drift in a different direction. So retention time replicate view is very handy in Skyline. You can also do some things with the peak area plots where this is peptide area view. So this is looking at all the peptides that are in your document. You can order them based on their retention time. And so the earliest should always be the earliest, the latest should always be the latest, and they should have the same relative order in between unless there are a couple that kind of co-elute and might flip-flop, and that's sometimes okay. And so what this is showing is over 10 replicates, the average peak area, and then standard deviation in those peak area measurements. So you want the peaks to be big and the error bars to be small. You can change the view to peak area CV, so it's the same data, but now we're just looking at the coefficient of variation, where this is 5%, 10%, 15, and 20. Anything above 20, in my opinion, is too high, and something's wrong. So you got to go look at it. Ideally, you want things to be as low as possible. And then if you click on a particular peptide and look at that over the course of injections, whether you did 10 all at once or once a day for 10 days, you can look at the absolute intensity, the stability of that. If you see one drop out or one spike up, then there might be some funny business going on, and you would want to go and look at that data. Oh, and I should point out, peptides that have lower peak areas, like this one, second to the end, often have higher CVs because it's a function of their abundance. So that's not out of the norm. So this was one of the reasons that we started evaluating system suitability in like a study-wise format. Was we had one lab that was running system suitability when we just started, and their later, so these are peptides ordered by retention time. The later eluting ones were all very low intensity, and we thought that was odd. And the early eluting ones seemed to mimic that of other labs. And when we changed the plot layout to looking at peak area CV, we noticed that about halfway through, all of these peptides had greater than 20% variability. Like, that's odd, especially when you see it as an effect of retention time. You're like, oh, something weird might be going on with the LC. And that helps us pinpoint if it's the LC or the MS. So after we pointed this out, and they're like, oh, what's going on? They had an exigent pump where you calibrate your flow, you measure your flow, and then you can calibrate it by inputting, like, I forget, you input a period of time that you're going to calibrate it over or something, and whoever did it put the wrong value by half for pump B. And so the acetonitrile that was being delivered over the gradient is low at the beginning, and pump B is working harder at the end. But it wasn't working hard enough, and so the peak areas for these peaks weren't as reliable because the peaks were broader and shorter. After they fixed it, after calibrating, you can see these peak intensities pop right back up, and their CVs were less than 10%. So if you can begin to look at your data and collect the information from your system performance, you can begin to troubleshoot things without having to call an engineer. So these are two examples of the longitudinal system performance where we're looking at one peptide over, and this is longitudinally from September 19th to September 29th, so over 10 days, and looking at the absolute peak area of one of our standards. The system suitability was run every 8 to 12 hours, so 2 to 3 times a day, and this looks pretty good. When we looked at another system that had it run from September 2nd to September 14th, so a little bit longer period of time, we noticed stable, 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 and then this gradual decrease. So remember that these samples were run every like 2 to 3 times a day. And so not only is that gradual decrease something that wouldn't totally be obvious until you plot it like this, but it might not be recognized until like the system is completely not functional. So what the operator decided to do here is like, oh, that looks like it might be related to the MS due to charging. So they took off the ion source, cleaned it up, popped it back on, and then the intensities went back up to where they were expected to be. Are there any questions so far? Okay. So there are other ways you can look at it besides Skyline, and some of the vendors might have software that do that. This is a metrics plot out of Analyst, if you're using a SciEx instrument, that's looking at peak area over number of injections. And so here you can define the average and then standard deviation or confidence intervals outside of which you would want to say like, oh, something bad is happening. You can do the same thing in Excel. Again, a little more labor intensive, but it builds character, right? So for people that are using different instruments like, oh, this is a QTOF, this is doing a targeted MS2 approach, so like PRM or Pseudo-MRM, where you're monitoring the fragment ions of a bunch of peptides in the sample. And this is how the system was set up, targeting particular mass regions over different retention times. And these are what the data show. This is one particular peptide that's displayed. The idea here is, though, so this is over a year. This was data acquired from the lab. And you can see kind of stable regions where the signal drops out. You can see the retention time over that time and the peak that was being monitored. So they targeted that data as well as doing data dependent at the same time. What they were able to do with that information is kind of test to see if looking at protein coverage from their data dependent run was a good indicator of system performance. So this particular sample, when searched, got, where's the percent coverage? 48% sequence coverage. This one that's about the same intensity got 46% sequence coverage. This one down here that's like half or a third of the intensity of the other two got 49% sequence coverage. Oh, we must be doing awesome if it's 49% sequence coverage, right? Kind of, but we're not, because the peak intensity is so low. Like, what are we sacrificing besides, we're obviously not sacrificing sequence coverage for that protein, but like clearly something is not quite right. So this is why looking at number of peptides or sequence coverage is not always the ideal and sole indicator of system performance. This is a flow chart I pulled from this paper that came out in 2007 because it's just a logical progression and thought process of what you would want to do. Set up the system, inject your samples. Is the system performing well? Yes, start your sample batch. No, then troubleshoot, and then start back at the beginning. So it's pretty logical, but it's also something that not everybody thinks of or thinks that they need to do. And I would say it is a very important thing. Okay, so a while back, this is involved in the evolution of system suitability evaluation for proteomics experiments. There wasn't much for proteomics experiments. And so Michael Bereman, who used to work in Mike McCoss's lab, now he's at NC State, he developed this thing called Statistical Process Control for Proteomic Experiments, or SPROCOP. It used to be in Skyline. Is it still? It is, okay. So the idea is you run your samples and process the data in Skyline, and Skyline will generate these control charts for the different metrics, whether it's retention time or peak area or full width half max. It'll give you the mean of the data and then set up 2 confidence intervals based on, I think it's 2 and 3 standard deviations. And when you see something go outside of those standard deviations, then clearly something is probably wrong. And then from those metrics will generate a Pareto analysis chart. And this gives you an idea of which of those metrics is responsible for the system going out of performance, which one is mostly responsible. So it breaks it down into the different categories. And so this is an example, tracking longitudinal data and plotting those. So you'll notice this has the same profile as this. It's just plotting peak area. It gives you the percent CV for that peptide. These are in control. There's some stuff happening here that seems systematically weird. And then it plots these control charts for all the different peptides that you would be monitoring for. And you can see if there is an effect of retention time on the CVs or anything else. So in summary, it's really important to set up your system with a good system suitability protocol. If you're starting, that's you're at the hardest part, but you've got nowhere to go but up, so give it a go. Don't waste precious sample and time without making sure your LC-MS system is performing. I learned this the hard way several times until I got it in my head, but you do a reaction with a sample and then you analyze it and you don't see the product. You don't see what you're expecting. You're like, oh, well, did the reaction go wrong? Maybe. Is your system performing right? If you can't say yes, then you don't know if it's a system or your reaction. And so don't waste your time running something if you're not sure the LC-MS system is working. Use system suitability to monitor changes over time. This by far has helped me to minimize my instrument downtime, and I've been able to learn things about how to keep it running well without having to call in service to have them fix something. Keep examples of poor system suitability data to help troubleshoot future issues. So the idea here is, like, if you accidentally injected air on your system because your autosampler vial was empty, you can look at the pressure trace, you can look at no signal, and be like, oh, and those things combined will help you know, like, I injected air. So you might not have to go through the entire troubleshooting process each time if you have, like, common things that are happening to your system, whether it's dirty or the solvents aren't degassed or you ran out of sample. And then here are a bunch of papers that I highly recommend if you need to, like, fall asleep on your plane ride home reading, because they're not super exciting, but they're packed full of information about how to do this, how to evaluate your data, how to set it up. Okay, any questions? Yeah, yep, I do that. So I started off with a system suitability standard. Originally it was something cheap that we just kind of put together, and eventually we realized, like, that's not as reproducible and stable as something we purchased that is QC'd by a company for stability. So I moved to the Pierce retention time standards. A similar one would be the Biognosis IRT peptides, and I used that as system suitability. Then, after a period of time, I was like, man, it would be so awesome if I spike these into my sample so that when I'm running my real sample, I have that extra bit of confidence that things are working okay. The responses of the peptides and their peak areas and retention times might not be the same as they are in your system suitability, but they should be similar. And so if it comes at a cost, it does cost money, these things, because they're qualified and stable and all that, it adds up. But if you can put that in your budget, it helps improve the confidence in your data that you're acquiring, both with your real samples and your system performance. Yes. Okay, so the question was, pretty much paraphrasing, can you use the internal standards that you're using for your biological study as an internal standard? So I was in a lab that did that, and everybody had their own biological study. And so when a new person would go up to start using an instrument, they would run their standards and be like, oh, it's looking good. And then the next person would go up and run their standards, and they'd be like, oh, it's looking good. Those people were responsible for tracking their own data and making sure the system performance was good enough. The way they did that was just by looking at the y-axis intensity, not really doing anything else. So the issue with using your standards is if you're the only one using that system and you only use it for that purpose, yes, by all means, you can track those standards over time. But if you're on a shared resource or if you're in a core lab, you want to make sure that there's something that has more data points in a longitudinal time frame so that you can see when things are not working quite right. Because when you look at a y-axis intensity and you see it go from 1.76 to 1.46, you might think, oh, no big deal, but that actually might be critical. That was a good question, because that's what we did initially, and then we realized, like, oh. Or if someone saw something going wrong and they didn't tell you, and then you start running, and then you see intensities go down, like you just wasted a bunch of injections on something that someone else could have told you if the same sample had been run consistently across. Because scientists aren't always the best at communicating. I don't know if you found that, but I have. Terrible. Yes?
If you use the RTs, do you just put in a lot of them, or do you maybe use a little less to test the sensitivity also? Or, I mean, also, if we just use RTs, we sometimes don't have trouble seeing the last one. And if we use a matrix with them, that one is more stable or so. Do you have anything like that?
Yeah, so I can tell you what I do. I don't know if it's totally right, but it's what I can defend. When I have a system suitability standard, I run heavy labeled peptides spiked into a sample matrix that is similar to the samples that I would analyze. So I use a lysate digest with heavy peptides spiked in. I target those heavy peptides and I track those over time on the triple quad. The purpose of the background matrix is if the system is getting dirty, you're likely to see the effects with presence of a sample matrix than if you're injecting peptides in solvent. If you're having isolation issues on the MS, you're more likely to see interferences if you have a matrix than if you're analyzing peptides by themselves. So and then as for spike level, so the sample matrix, I don't want to blow through sample, but I also want it to be somewhat representative of what I'm normally analyzing. I normally inject a microgram of my sample matrix on column. For a system suitability, I might use anywhere from 200 nanograms up to a microgram, depending on how much of that matrix is available. If I'm like growing cells myself and lysing them and I have gobs, then I might opt for the 500 nanograms. If I'm buying cell lysate digest from a company and it's super expensive, I might go on the lower end, but I keep it within an order of magnitude of my range. And then for the things that I target, I would try to figure out, I would initially run spikes to see what's the lowest level I can spike into my sample that still has at least 20%, if not better, variability in peak area and retention time. And then I would be in that range. So the idea is if you start seeing your sensitivity degrade, those peak areas would go down and your percent RSDs or CVs would go up. And that would be a good indicator that you're on a threshold of the sensitivity and like something might be dirty or something would need to be fixed. I wouldn't spike in hundreds of femtomoles because that's not going to tell me much about the system. And then you had mentioned if you're monitoring a series of peptides, sometimes you might see one at the end and sometimes it doesn't show up. We often blame it on the sample, like, oh, maybe that peptide's not stable, but sometimes these things are dependent upon system performance. So for the Pierce retention time standards, the earliest eluting peptide, if I use a trap column, I don't always see it, but it's dependent upon my loading flow rate. So if I load super fast, 10 microliters per minute for a short period of time because I want to get through the run, I might not see that peptide because it just blows through the trap and it's gone, which means if I do that for my real sample, other peptides might be missed too. And so I try to set up the method similar to how I would normally analyze samples. It might be a shorter run so I can like get more in there without having a huge overhead in time, but trying to just monitor those peptides. And then if I see one that sometimes disappears and sometimes appears, keep an eye on it and try to figure out why, whether it's been in the vial too long and it's falling apart, it's a bad sample, or maybe there was too much acid in the sample, or maybe it is a chromatographic issue that we're seeing. Yeah. Does that answer it? Okay. Anybody else? Yes.
When you say find your own kind of QC, and what parameters you want to keep in your kind of tracking file?
I think. So AutoQC has a series that it automatically looks for. If you're not using that, these are the ones that I would keep track of, because things like peak shape and full width half max can be related to your column and LC and the connections. If all of a sudden you're seeing carryover when you shouldn't normally, that's another thing that would be more LC indicative. So these are all metrics and numbers that can be pulled out of Skyline or out of data processing software that you might normally use. Some of it might not be automatically calculated for you, like not all software will tell you the full width half max, but you can go into the chromatogram and measure what you can measure it. There's a question over there. Yeah. How do you predefine if you haven't defined any yet? You have to evaluate a good amount of data and then set up your confidence intervals. And so that's why using software like SPROCOP or AutoQC, I think, has it in there. You can, if you're starting with nothing, make a bunch of measurements. Don't just rely on 10 injections one after the other. Like, do that, but then do it once a day for a number of days. And then once you get enough data to be able to confidently define your acceptance criteria, then you're good to go. But when you're starting out, it is a little like, oh, I don't know what's okay or not. So you just kind of look at the performance of your system and see what it tells you. And keeping in mind that the smaller variability, the better, right? If you see peak area variability of 20%, you always see 20 or 30%, like that's not good, right? So some of it's common sense, like, you know, keep the variability low, but also see what your system's capable of. Yes?
So this is more instrument specific. You cannot really have compare. You can have one method, but you have quite a few instruments.
Yeah, you can run the same method on a variety of instruments, but the data those instruments provide shouldn't be lumped together. You can compare data sets, and I mean, intensity and peak area is an arbitrary value depending on what vendor system you use. Some of them multiply it by numbers, some of it's counts per second. So the units and the absolute magnitude can't really be compared, but the trends can, right? You should have similar metrics of reproducibility. And retention time should be really similar. Of course, it'll vary depending on column stationary phases, but retention time, not absolute retention times, but reproducibility or retention times, full width, half max, peak shape, those things should be comparable.